Guys, here's a video of the notes on the topic heterochromatin, its types, organization and formation and significance. So the region of the chromosome which remains darkly stained during the interphase as well as distinct phase is called as the heterochromatin, and it's transcriptionally inactive and is more densely packed. It is generally found in near the centromere and telomeres, and it has two types, that is consecutive heterochromatin and the facultative heterochromatin. So here is a difference between the consecutive and the facultative heterochromatin. So consecutive heterochromatins contain the satellite DNA, also known as the SAT DNA, and while in the facultative heterochromatin it has line, that is long interpersed nuclear element repeated sequence. So here is the figure of the heterochromatin. So the apical part of the chromosome known as the telomere, and there is a dark band and the light band known as euchromatin and the heterochromatin. We can say that the euchromatin is the active part of the gene, while the heterochromatin is the inactive part of the gene. So heterochromatin formation, how the heterochromatin formed. So it is formed by the deacetylation and the methylation of the histone protein, that is H3. So HP1 protein, that is histone protein 1, is the key protein in forming the mammalian heterochromatin and acts by binding to the methylated histone H3. So here are the step-by-step process of the formation of the heterochromatin. So this is a part of the active chromatin. So by the help of the deacetylation by histone deacetylase enzyme or the histone acetylase transferase, that is HAT, the removal of the acetyl group. So AC represent the acetyl group. So removal of the acetyl group process known as the deacetylation, and then the methylation by adding the methyl group by an enzyme known as the histone methyl transferase, that is HMT. So recognition by the chromodomain of HP1. Then here this is the part of the chromatin, and with the addition of the methyl group. Then next step is the binding of HP1 protein by their chromodomain is methylated, that is H3K9, that is initiation of the heterochromatin, and then the propagation of the heterochromatin by self association of the HP1 and process known as the heterochromatization. The methylation of the H3 at lysine 9 H3K9 demarcates heterochromatin, while H3K4 position demarcates the euchromatin position. So here the formula: K represent the lysine position and H3K means lysine and at position 9. So the S Bombay, the H3K4 and methylene 3 is refers to as the lead-2 interaction with the LR4 H3K9 methyl transferase, resulting in the H3,4 hypomethylation and the heterochromatin formation. Here is the term RNA interference, which is a silencing gene, fundamental gene silencing pathway which target the mRNA and break down into small pieces and prevent from the translation process. So here is a chart. The double-stranded RNA cleaved into shorter fragment with the help of the Dyson enzyme into the siRNA, shRNA and the miRNA, that is microRNA, short hairpin RNA and the small interfering RNA. Then these three RNA is regulated by the Rex factor, that is RNA-induced silencing complex, which cleaves the mRNA and then the gene gets silenced. So in this pathway, so here the figure, the double-stranded RNA with the help of the Dyson enzyme, it get fragmented, and then the gene silencing enzyme known as the Rex factor, slicer and the Argonaute 2 known as Rex factor get attached with this single-stranded RNA and the gene gets silenced. Next topic is the heterochromatin formation in the centromere region. So the CNPB is a centromeric specific protein found in the human cell, is required to initiate modification of the H3 by deacetylation of the K9, that is lysine 9, and the K14, followed by the methylation of the K9 that triggers an association with the HP1 that leads to the formation of the heterochromatin in the centromeric region. So this figure signifies the position of the telomere euchromatin pericentric region, the SAT DNA, also known as the satellite DNA sequences. So significance of the heterochromatin. So it maintains the structure integrity at at centromere and telomere. It appears to be involved in controlling the transcriptional validity of the genome. The constitutive heterochromatin contains sad DNA are able to fold on themselves and may have important role to the formation of the highly compact structure. These are of a great important role in disease control. So this is all about the heterochromatin, its types, their significance, and the formation and organization of this heterochromatin. Thank you.